All right, hello. So this is a re-recording of chapter 13 presentation for Biology 241. And sorry, my dog is deciding to stretch and tap dance right now because I started recording, so he has to make lots of sounds. That's how it works. Okay, so this is chapter 13. So now that we've learned about things like neurons, glial cells, CNS versus PNS, and other general nervous system information, it's time to start getting more specific. And so that is what we're going to do here. So we're going to talk about the spinal cord, its cross-sectional anatomy, its physiology, its functions, and also the spinal nerves. And then we'll kind of delve into their role in things like reflexes and a bunch of other fun stuff. So as we can see in this illustration, we've got the spinal cord and some little spinal nerves coming off of it. And what you can see is that in addition to the spinal cord, which I'll point out right here, so it's this middle white thing, we also have a bunch of spinal nerves which come off of the spinal cord and then branch into the dorsal and ventral ramus, as you've learned in lab. But we can also see these braid-like networks of neurons coming off of the spinal cord. Let me try and pick a good color to do that in. So here, for example, here, here, and here. And so all of these things each of them individually is a nerve plexus. In plural, there are nerve plexuses. So plexus means a braid-like network of something. In this case, it's a braid of nerves. So these are places where the spinal nerves have a more intricate branching style because they're going to innervate a particular location or place. So that's kind of the deal there. And we'll have just a look at each of the plexuses and what they're for. But in general, overall, the spinal cord and the spinal nerves, respectively, provide a pathway for impulses, which means, let's paraphrase this, shall we? Route for action potentials. And we're going to make note that this is afferent and efferent. So that's thing number one. They also mediate spinal reflexes, which is one of the things that this chapter is about. So a spinal reflex is one which is coordinated and executed by the spinal cord. So the information about what's happening is sent up to the cerebrum, but the actual reflex and response can be coordinated by the gray matter of the spinal cord all by itself. So sorry, one second. So mediation of spinal reflex is another one. And one of the sort of little subtexts of the spinal reflex mediation, this really probably should be 2A instead of 3, is integration of sensory and motor impulses. So that means in nervous system speak, integration refers to taking sensory information, doing some computation about how that sensory information pertains to homeostasis, environment, the health and well-being of the individual, and then using that information to do some motor function. So that could be muscular, it could also be glands or other effectors, it really just depends. But that's what integration means in this context. So with regard to the gross anatomy of the spinal cord, meaning just big stuff that we can see easily, after the medulla oblongata, so remember the brainstem includes the pons superiorly and the medulla oblongata inferiorly, and that's the last part of the brain that is technically quote-unquote brain. And then the medulla extends down into the cervical spine and all the way down to lumbar vertebra number 2. And if you're thinking two, but then why do the sacral vertebrae have holes in them and what's the deal with that? Well, after the lumbar enlargement, which is an area associated with the lumbar plexus, what we have is this little cone. So the spinal cord comes to a conical shaped tip called the conus medullaris, which means cone of the medulla. So this is the end of the cord, so it truncates at L2. However, it doesn't stop. Instead, it just stops being a solid rope or a cord of nervous tissue, and instead it branches out into a multiple nerve structure, which is called the cauda equina. And the cauda equina means tail of the horse. And as you can see, because it's multiple nerves traveling in parallel and then kind of branching off, it kind of looks like a little horsey tail. So these are the spinal nerve roots that basically descend through the lower portion of the vertebral canal, which is the tube formed by the vertebrae stacking on top of each other. So between L3 and L5, so the remaining few lumbar vertebrae, and then continue on down into the sacral spine. So finally, we've got the phylum terminal, which means end filament. And this is a specialized extension of the pia mater that functions to anchor the spinal cord to the sacrum. So your spinal cord can move and flex with your spine, but it does so best and most healthily if it is contained in and also anchored to both ends, so both the cerebral end and the coccygeal end. So, interestingly, the phylum terminal associates with the coccygeal ligament, which is a ligament, so a connective tissue structure associated with the coccyx. There is a condition, which I don't remember the name of right now, escaping me, but there is a condition in which the phylum terminal has too much tension, which tends to cause neural problems for the people that suffer from it, which is interesting. So that's what the phylum terminal is, and that's a pathology that's pretty unique that can result from it being too taut. So now we're going to look at the segmental anatomy of the spinal cord, which is another way of saying a spinal segment or a spinal cord segment is just if you took a cross section of the spinal cord at a particular level. So is it in the cervical spine? Is it in the thoracic spine? Is it in the lumbar spine, etc.? What would you see in cross section? What histological features and gross and anatomical features are there, and what do they mean or do? So there are 31 horizontal segments, and this 31 is going to just correspond to the spaces between the vertebrae. Remember the intervertebral foramen from lab that can only be seen from the side and only in an articulated spine, where the spinal nerves come out. There are more of those than there are vertebrae, and of course that's because there's more spaces between. So the horizontal segments are where the spinal nerves enter and leave. So we refer to these segments as, C4, T8. So they have the corresponding number and letter relative to the vertebra that's nearby them when they come out, essentially. So it's important to remember that the spinal cord is formed by the fusion of the dorsal root, which remember from lab, we've talked about it. The dorsal root is carrying afferent signals. which means sensory information. By contrast, the ventral root is carrying efferent signals, which is to say motor commands. So the spinal nerve is then going to be necessarily a mixed nerve. Let's add some green to that, but I'm going to do it in this. So the spinal nerve is carrying both afferent and efferent, both sensory and motor information. So if I were to ask you in a test, true or false, each spinal nerve carries only ventral or only afferent or only efferent information, you would say false. No, it carries both. It's the dorsal roots that are separated by that function. So that's kind of the deal there. So let's just draw some little arrows. So we know that action potentials are moving this way through the dorsal root, and they have to pass the dorsal root ganglion to get there. And the ventral root provides information coming from the CNS away, and then the spinal nerve is both. So you've got action potentials in both directions. So the analogy, obviously, is the roots are a one-way street, and the spinal nerve is a two-lane in opposite directions road. So that's pretty easy to understand. The dorsal root ganglion, which you might notice if you haven't looked at this closely before. I know my students have, but if you're a student in the future, or you're just a rando who shows up to my YouTube channel for some reason, welcome. You'll notice that there is a dorsal root ganglion, but there is not a ventral root ganglion. So in order to understand this, we have to consider a couple different things. I'll list them for you. So number one is that most sensory neurons are unipolar. And if you're not remembering quite what that means, we talked about the structural types or shapes for neuron types. It's the one that looks like this. So in this arrangement for a neuron, the soma, the body, is off to the side on a little stalk, kind of like a lollipop on a stick. There is a very small end that is dendrites. And then the initial segment of the axon is right next to the dendrites. And then the rest of it is axon. So that brings me to the second thing that we have to remember, which is that everything outside of the spinal cord, so anything that's not spinal cord or brain, is automatically peripheral nervous system. And there's some particular ways that we refer to groups of cell bodies or groups of myelinated axons, depending on whether they're in the central nervous system or the peripheral nervous system. So since the dorsal root ganglion is a ganglion, it makes sense to note that ganglion equals a group of neural cell bodies in the peripheral nervous system. So it doesn't make sense to position your cell somata, which is the plural of bodies, all up and down a nerve. You kind of all want to put them in the same place. So they get centralized, and that centralization, that centralized location, is a ganglion. So that means that what's in here is just lots of these all together. So now let's compare that to the ventral root. So again, white matter is what we refer to as tracts or groups of myelinated axons traveling together in the central nervous system. Gray matter are groups of cell bodies and glial cells in the central nervous system. So those are the equivalents of nerves and ganglia, but the CNS version, because they're arranged a little bit differently, but they have the same kind of basic substructures. So if I tell you then that the cell bodies of lots of different kinds of motor neurons are sitting there in the ventral root, or excuse me, the ventral gray horn of the spinal cord, and that they project their axons out like so, we don't need to have a ventral root ganglion because the cell bodies are positioned inside of the spinal cord. They're not outside. So no bulge, no ganglion for that reason. So that's the reason why you see a dorsal root ganglion and not a ventral root ganglion. There is only a ventral root. So hopefully that makes logical sense to you. Okay, so moving on, we're going to talk about the spinal meninges. So singular is meninx, which is fun to say. So I also want to point out that the cerebrum has the same sets of coverings. So we'll kind of circle back to this eventually. But it's the same meninges in the entirety of the central nervous system. So these are, I'll talk about meningitis in a minute, the dura mater, which is dense irregular connective tissue, and it is extremely tough. And in fact, dura means tough, and mater means mother. So it's not matter with two T's, it's mother. So the meninges are the three mothers, the mothers of your brain and the mothers of your spinal cord. They protect, they comfort, they cushion, they cradle like mothers. So the dura mater is incredibly dense and fibrous, dense irregular connective tissue, and it is very, very intense stuff. So it's so dense that it's kind of like Tyvek, which is that stuff that they wrap homes that are being built in. If you've ever tried to tear Tyvek with your hands, you can't do it. It's also extremely puncture resistant. So it's very thin material-wise, but it's extremely difficult to either puncture or tear. So it's very resistant to shear forces. And it's the same thing as the dura mater. So the arachnoid mater is the spider mother. So this is a mixture of collagen and elastic fibers in a thin arrangement. So it's not as tough as the dura mater, but it's also not itself insubstantial. And this is made of a mixture of collagen and elastic fibers, like I said, and it's kind of a little bit stretchy, so it provides flex and the ability to flex while being protected. And it's got little denticulate ligaments that reach down uh, across the subarachnoid space, touching the final mom, which is the pia mater. Pia means soft. And this is the one that actually touches the neural tissue of the CNS itself. So on surface of brain and spinal cord. So it's a thin, much looser connective tissue layer. And so these three layers are the non-bony protection, and they function to do a bunch of different things. One of them is to isolate the CNS tissue from the general bloodstream and general extracellular fluid. And that's important, right? Because we know that we use the brain and spinal cord, our central nervous system, we use it, oops, sorry, I switched positions and PowerPoint decided it wanted to as well. So we use our central nervous system as a control center, and that's also where our personality and our self lives. So kind of don't want that to just get infected with, whatever is floating around in our extracellular fluid by accident. So that's one thing that it does. The arachnoid mater in the cerebrum is also involved in recycling of cerebrospinal fluid, and it provides a containment for those fluids so that as the cerebrospinal fluid is made and circulates, it's not only picking up wastes, but it's also another physical barrier against damage to the central nervous system. So lots of good and fun benefits there. Speaking of spaces and fluids, similar to how there's an intervertebral foramen, but it's only something that you see if there's two vertebrae stacked on top of each other, there are also meningeal spaces, which are only present between meninges, or around them at least. Oh, really quickly, I forgot about meningitis. So itis equals inflammation, and menin refers to the meninges or a meninx. So meningitis is a bad, potentially life-threatening inflammation of the meninges. And remember, inflammation produces swelling due to histamine and heparin, promoting the leakage of plasma out of the blood vessels via widening them and thinning the blood. And swelling can push on the central nervous system tissue and cause an interruption in nervous system function in the place that's being pushed on. So meningitis is really a description of the disease set of symptoms. So it's describing the condition in which the meninges are inflamed. But that's not the same thing as the etiology, which is a word you're going to hear me say more than once. So I'm going to take a moment to explain the difference because I think it's important, and it's also something that a lot of, so the layperson does not understand, and it prevents people from understanding diseases and pandemics and disease processes uh, in a way that could help them be healthier or advocate for themselves better at the doctor. So I think this is important general knowledge in addition to being important for my students. So let's talk about, I'm going to use meningitis as an example, and then I'll give you another one. And of course, right off the bat, can't spell. So I already described meningitis as an inflammation of the meninges. So remember, we have our cardinal signs of inflammation, which is redness, not that you'd see it, swelling, heat due to increased blood supply, and of course, pain. So inflammation is predictable. It's the same set of things across all the tissues that it affects. This just happens to be that set of things affecting the meninges. That's what meningitis means. Now, notice none of that information contains any details or indication of what caused the inflammation. We're just saying, hey, the meninges are inflamed. So, I mentioned the word etiology. And I should make that O and that L a little bit further apart so you can tell it's not a D. Okay, so etiology is the cause of a disease. Also, maybe we should take some time to define disease a little bit. That's worthwhile. So, diseases or pathologies are interruptions to physical homeostasis that are nameable because they follow a specific pattern. So that follow a pattern. And usually the pattern is consistent or relatively consistent symptoms. So obviously not every disease manifests in exactly the same way in every person, but the way we decide what disease someone has diagnostically is by seeing what list of symptoms they have and drawing a conclusion based on that. So, if etiology is the cause of a disease, and the disease is interruptions to homeostasis that follow a pattern, then if we revisit the definition of meningitis, we still don't have any information based on this about what's causing the meningitis. So this is just the name for uh, inflammation of the meninges, which would present with consistent symptoms. So the etiology of meningitis has a few possibilities. So we have two types overall: bacterial meningitis. So this is caused by bacteria. And two is viral meningitis, which is, of course, of viral nature or origin. Bacterial meningitis is grosser because it ends up creating pus between the meninges, which I know, disgusting. You can, if you're not squeamish, you can look up pictures of people with bacterial meningitis who've died of it and their brain autopsy, and you'll see the pus. It's real gross. However, viral meningitis is often more difficult to treat because antiviral drugs really just target viral replication. They don't kill the virus in the same way that an antibiotic would kill a bacterium. So viral meningitis can be much harder to treat. So let's talk about those symptoms, the symptoms of meningitis, especially since I know many of my students have children, and children, college students tend to be the groups of people that are at most risk of meningitis, apart from the highest risk group, which are intravenous drug users. But intravenous drug users are at higher risk for a whole bunch of stuff disease-wise. So the most frequent pattern of meningitis outbreaks is among college students who live in dorms together and have a lot of contact with each other, or in daycares. So be watchful. Okay, so let's describe the consistent pattern of symptoms that would lead a clinician to determine that the disease being experienced by somebody is meningitis. So, one cause of meningitis is, or we not cause, symptom is stiff/sore neck. So, neck pain is a big one. Now, don't panic if you have neck pain. It has to come with these other little details. So, other one is deep fatigue/sleepiness. And vomiting, usually without any nausea, which is unusual, right? Usually we vomit if we're nauseous, or nauseated, excuse me. But in this case, because the etiology is different from stomach irritation, we're going to end up with projectile vomiting that is not associated with nausea. So just suddenly you're barfing. And then, of course, also a fever. So, and this is usually a high fever, which also endangers the brain. So if you see this combo of symptoms, that is an indication that you should not delay. You should just go to the hospital and let them figure out if you have meningitis, because if you do, the likelihood that you'll die without treatment of it is pretty high, or whoever the meningitis sufferer is. So that's the difference between a name for a disease and the etiology of that disease. So the other example I'll give you, which actually comes from real life for me, is the following. So I'm going to write the disease name, pneumonia. I don't know why it keeps doing that, which is fluid buildup in lungs. And it's usually caused by a pathogen of some kind irritating slash inflaming lung tissue, or sometimes the immune response to a germ. So your immune system will often create fluid buildup, a la inflammation, when it's trying to fight something off. So pneumonia is the disease, and it's described by this. So symptoms include shortness of breath, coughing, etc. Here's the thing, though. This does not tell you anything about the etiology. So the thing that in my life that applies to this, and this is where I got this example, is my parents' neighbors. So my parents' neighbors are nice and elderly and also extremely conservative to the point of having a hard time distinguishing between news that is real and relevant to them and false information being circulated on things like Facebook or Fox News. Um, so, because they can't do that, and they don't have any medical training, so why would they understand this difference? They wouldn't. The husband got COVID because they wouldn't get vaccinated, and they also didn't want a mask for personal or political reasons, what have you. And so the husband got COVID and ended up being hospitalized for it, which at his age is very scary. So when my mom told me that their neighbor was hospitalized, I was like, oh, well, do they believe in the coronavirus now that he has it and has been hospitalized? And she said, oh, no, not at all. And I was like, how can that be? That is mind-boggling to me that you could get sick with an illness and still be like, nope, not real. Here's the thing. The wife responded, he doesn't have COVID, he just has pneumonia. Among the causes of pneumonia are bacteria or viral infections, kind of like meningitis. So, can COVID cause pneumonia? Absolutely. That's why most people that have died of COVID died, is because they had pneumonia that was so intense that it was essentially untreatable. So, on their autopsy report, it would be written that their proximate cause of death was suffocation due to pneumonia, but their ultimate cause of death was the coronavirus that caused the pneumonia. So that's another example of a disease name versus etiology confusion or mix-up that, in that case, has the potential to cost someone their life. Fortunately, elderly parent neighbor recovered and he's fine, albeit, frustratingly has not updated his or his wife's. Neither of them have updated their opinion or information based on their experience, which is confusing to me, but whatever, to each their own. But this is science class, and I'm going to tell you science facts, and those are the facts. Okay, so meningeal spaces. So these are just spaces between or around the meninges, easy peasy. And this is another thing where if you memorize the order of something, it's easier to get to the right answer even if you're initially not sure. So we can also use our friends' root words to do this. So epi means superficial to, and then dura refers to dura mater. So this means outside of the dura mater. Now, in the spinal cord, this is possible. In the cranium, this is not. So this is one of the differences between the meningeal arrangement between the spinal cord and the cranium. I'll explain what the cranial situation is later when we get there, but for now, in the spine, it is a fat-filled space, and it also contains blood vessels. And it protects the spinal cord. So one of the things that fat does, it's excellent padding. So it's kind of like the packing peanuts of your body. Which is nice, right? And so fat gets a bad rap, but fat is really important, and it's so important that you need it to do things like pad stuff, but also make hormones. You'll learn more about that next term. So I'm just going to go ahead and give a nice plug for reframing how you think about fat, because it's not like inherently evil. It has a very important set of functions. So the epidural space is also where epidural anesthesia is administered. So when they do that, there's a cannula and it goes in there. And then the cannula releases anesthetic, which bathes the epidural space, and that filters into the subarachnoid space and bathes the nervous tissue in anesthetic, which deadens the nerves, a la epidural anesthetics. The subdural space is a really, really thin space. It's technically a potential space, so it's just that the arachnoid mater and the dura mater are not, they're up against each other, but they're not connected to each other. And this should say ECF, it's not intracellular fluid. And then finally, the subarachnoid space, which is under the arachnoid space, or layer, so arachnoid mater, and then down from that is the subarachnoid space. This is filled with cerebrospinal fluid, and the cerebrospinal fluid absorbs shock, it protects the spinal cord. I think it's also important to note that it removes, wastes, delivers nutrients, etc. So, when our central nervous system tissue produces waste products due to it being busy and producing lots of like neurotransmitter breakdown products, metabolites, etc., that has to go somewhere. So where it goes is into the CSF, and then the CSF ends up being dumped back into the venous circulation eventually. So more than just shock absorption, but that is a really good function. Okay, so let's talk about the gray matter arrangement of the spinal segment. So you can see that the gray matter itself is arranged into this pretty lovely butterfly shape, and the wings are made up of horns, and the middle part is made up of commissures. So I've discussed this before, but a commissure is an area where decussation happens. And decussation is defined as crossing over of a neural signal from one side of the body to another. So, what does the gray matter contain? Well, it contains the same stuff as the gray matter of any other central nervous system area, which are somas. Somata, that's the correct pluralization. And remember, somata are just neural cell bodies, and these are organized into groups according to their function. So I'll explain those in a minute. And then also unmyelinated fibers. So if interneuron, which is a neuron that connects to others or what have you, if the axon is relatively short, meaning that the action potential has a short way to travel, it's not always necessary to myelinate them. So sometimes they're not. Okay, so if we go to this division over here, this is where same Dave comes in. So Why don't we remind ourselves about our nice, friendly, same Dave acronym? So this is an acronym to remember both the meaning of afferent and efferent and also how that relates to the spinal cord. So sensory equals afferent information, motor equals efferent information. In Dave, dorsal equals afferent and ventral equals efferent. So what that means is sensory information is coming into the spinal cord via the dorsal root, and therefore the dorsal or posterior gray horn is going to contain groups of neurosomata that are going to be responsible for receiving and processing sensory information. So this is what I mean by sensory nuclei. A nucleus in the central nervous system is a group of cell bodies and unmyelinated axons. And then we have somatic senses, so skin, proprioception, pressure, etc., pain, other things, temperature. And then we have visceral sense, which are any sense that mostly you don't get to feel or don't rise to your conscious attention. So, for example, uh, my brain knows generally what my pH of my stomach is at. It also kind of has some idea about how much oxygen or carbon dioxide is in my blood or my cerebrospinal fluid at any given time. It knows about the pH of my body fluids as well, and a whole bunch of other good stuff, including my blood pressure, my heart rate, yada, yada, yada. So none of those are senses that rise to my conscious attention. I'm not walking around on the earth being like, wow, my blood pressure sure is, 119/80 or whatever. Or I'm not like, it's really cool to have a blood pH of 7.5. I'm glad I have it. But rather, these are things that the sensory nuclei are sending up to the brain, and the brain is like, okay, so this is the information we need to do our job and keep the person alive. So we're going to do that. So they're part of feedback loops rather than conscious sensation. And then the same deal for for visceral and somatic motor nuclei. Somatic means voluntary skeletal muscle, and visceral is everything else, all the other effectors besides skeletal muscle. So that's the breakdown of these nuclei. So here's just a recapitulation of that same information in word form. I just like to draw it onto the picture because that's what's more natural for me. But for those of you who love lists and that's how your brains like to receive information, this is the deal. I do want to point out, though, that the word integrate comes back. And this is a word you're going to see a lot. So remember, integration is the processing of sensory information and it being made sense of or having something done with it by the central nervous system. So it's a shorthand way to say neurons talking to each other. Okay, so white matter is organized into tracts. And I think I would be remiss if I didn't mention or remind you of the definition of tracts, which is groups of myelinated axons in the central nervous system. So because the spinal cord segments have bilateral symmetry, they're all paired. So there's 3 columns, which makes sense because this is ultimately a cross-section of a column, and they are paired. There's one per side, and these are named things like anterior, posterior, and lateral, which is nice and easy because that just describes their physical location. Additionally, if we look right here, so we've got the gray commissure on either side of the central canal, but we've also got this one white commissure over here, and that's going to connect the left and right white matter. So taking a closer look at the columns, remember when we discussed the histologic organization of nerve coverings? I mentioned the word fascicles, and we use this word also in the central nervous system, but in a little bit different way. Um, Tracts are also called fasciculi, and these are bundles of fibers that have a common origin or a common destination. So they're all traveling together because they're coming from or going to the same place. Oopsie. So, as it happens, the tracts of the spinal cord, both ascending and descending, it's not as though they stop abruptly and then the brain has separate ones. They're all continuous because the continuity of information flowing into the central nervous system and up to the brain, or flowing down from the brain through the spinal cord and out to motor effectors, it has to be continuous. And depending on whether you're doing a motor thing or a sensory thing, most of the time that's going to include between 2 at the minimum and 3 at the maximum total neurons. And then processing occurs in the cortex with more of them, obviously, but the actual sending and receiving bit is going to be 2 or 3 neurons usually. We'll learn more about that in chapter 15. So obviously, going back to our same Dave definition and a mnemonic device, sensory tracts are ascending and motor tracts are descending. So returning to spinal nerves, remember they're paired, so bilateral symmetry results in us having one spinal nerve per side for each segment. And these run between the central nervous system and the periphery, but I want to tell you a little bit about this word periphery because it doesn't quite mean what most people assume it means. So this includes sensory receptors. And when we think of senses, most of us, if we're, not neuroscientists, we think of the 5 senses or skin sensations. And that is one kind of sensation, but there are other kinds. So we can divide sensory receptors into exteroreceptors, which means they sense things outside of the body, so stimuli coming in from the environment. But there's also oodles of oops, tried to do capital R for some reason, receptors. And these are going to detect internal conditions. So that's still considered the periphery because it's the very edge, not of the body, but of the nervous system. So that's kind of the difference. I find it's helpful to be explicit about that because otherwise the word periphery, people just kind of assume that it has the same meaning as it does in common spoken English, and it's not true. So again, spinal nerves are mixed nerves. They contain both afferent and efferent fibers. And as per, again, our same Dave mnemonic, the ventral root carries motor information only. The dorsal root carries sensory information only, but the spinal nerve is mixed, and I already explained the somas of sensory ganglion and why we don't have a ventral one. Okay, so I'm going to end this particular video on this slide just because I need to sort of keep the videos shorter so that they're easy to upload, and we're already at 49 minutes for this one. So cerebrospinal fluid components, I mentioned waste earlier. So the cerebrospinal fluid looks a lot like plasma, but because of the blood-brain barrier that's maintained by ependymal cells and astrocytes, there are some differences. So there's a few white blood cells that just, go in there and just check things out, make sure you're okay and your neural tissue is okay. Unsurprisingly, lots of ions. Why? Well, because action potentials. And there's also glucose, so this is food for neurons. Proteins of various kinds, lots of functions. Lactic acid, which is a product of glycolysis. and urea, which is a nitrogenous waste resulting from the breakdown of proteins, which is essentially equivalent to neuron poop or pee. It's the result of a neuron being metabolically active and producing a waste product as a result of that. So for us, as a whole organism, the result of being metabolically active and producing waste as a result of it is poop and pee. And you'll learn all the details about how those are made next term. But on a very tiny scale, a microscopic scale, urea and a couple other waste products are technically neuron poop. This is why we need to recycle and have our cerebrospinal fluid flow so that we can clear the neuron poop out so it doesn't end up poisoning our neural tissue. So let's take a look at, I mentioned the epidural, and I'll talk a little bit about that as well, but this is a picture showing a lumbar puncture. So we can see after the spinal cord, so here's the conus medullaris ends, now we're in the cauda equina territory. And so lumbar punctures are performed down here because the nerves here are covered in perineurium, and so they're more protected naturally. And also, if you use a needle or a cannula with a blunt end and the hole at the top, what that means is if this blunt end runs into those nerves, they're just going to kind of move out of the way. So we're not going to accidentally stab the spinal cord, which would be terrible, and we're not going to actually accidentally puncture any critical spinal nerves. We're just going to poke the little cannula in there and sample some fluid from there and take it out without potentially damaging anything. So this is why we don't use this, and instead we use, let's make this green, this guy. Huzzah! And the similar thing is used, this kind of atraumatic cannula or needle is used in epidural anesthesia as well. Again, same reason we don't want to like accidentally stab through the dura. So that's why a lumbar puncture is performed at the location that it is, and why epidural anesthesia is performed using a specific set of components, so not a needle actually. So when a lumbar puncture is performed, what they're looking for is this stuff normal? So there are normal ranges for all of these things in the cerebrospinal fluid. So a lumbar puncture is going to look at that. So if you were wondering, when I was talking about meningitis, how they figure out the etiology, so is it bacterial or is it viral? The answer is usually lumbar puncture. So can take CSF via lumbar puncture to assess etiology. Neat. Unfortunately, this is an extremely uncomfortable procedure. It is not fun, it sucks, so try to avoid having to have one if at all possible. Okay, so we're at 53 minutes. That's long enough for one video. I'm gonna stop, edit this really quick, and get it uploading so that I can get to work on the next video. So thank you for your attention, and I will see you in the next video.